Now we are going to see pathogenicity in infected person. What is the meaning of pathogenicity? So once the HIV virus enters into one person's body, how it is multiplying? That is only the pathogenicity. Pathogenicity means entry of microorganism and multiplication of microorganism in the infected person. That is called a pathogenicity. Now this is the macrophage. Just before we have studied, they are present inside the macrophages. Macrophage, white blood cell, it is our white blood cell. One HIV is coming and touching this macrophage. So surface to surface, they are touching each other. Then the HIV is transmitting only the viral RNA into the macrophage cytoplasm. Now how many strands of RNA will be there? 2 strands. Both the strands will come inside. So viral RNA will enter into the cytoplasm of the macrophage. Now in the cytoplasm of the macrophage, now it is what the viral RNA will do. They will be changed into viral DNA, many copies of viral DNA. Now that is the property of HIV. Now in the human immunodeficiency virus, RNA can be changed into DNA, DNA can be changed into RNA. So that is easily they can make it. So the viral RNA inside the macrophage will be converted into viral DNA. Enormous number of viral DNA will be produced. Now the viral DNA incorporate, that means joins with the host DNA. Host is the macrophage. It is having its own chromosome. It is having its own DNA. Both will incorporate. As a result of which, once they are incorporated, the viral DNA is making the cytoplasm of the macrophage to produce virus particles. Virus particles means new viruses. Once again, what will happen? Outer envelope, then inner capsid, then 2 strands of RNA, everything will be there. That lot of viruses, spherical shaped viruses will be produced by using the cytoplasm. So the cytoplasm of the macrophage is used. So the macrophage will die. Then once the macrophage, inside the macrophage, viruses are multiplied many, the macrophage, the dead macrophage will burst open and it is released. Okay. Now all the viruses are released. Again the viruses are entering into helper T cells. Now the viruses which are released from macrophages will go to the T helper cell or helper T cells, and they are once again the same process. The RNA will be injected, then new viruses will be produced, so the T helper cell will die. If the T helper cells are dying, then the immunity level will go down. This is the way our white blood cells are gradually destroyed, so our immunity level is gradually decreasing. In the lower classes you might have studied, so fever, mild fever will be there, but that fever will be there for more than a month. Diarrhea, there is loose motion will be there, that also for 3 weeks, 4 weeks, like that continuously. Weight loss, body weight loss will be there because of diarrhea, definitely body weight loss will be there. Decrease of immunity level, automatically I told, because the helper T cells are destroyed, so immunity level is going down. Many other pathogens, now not HIV is not killing the person, HIV is destroying only helper T cells, so immunity level is going down. Other pathogens like tuberculosis bacteria, okay, typhoid bacteria, okay, like that many other microorganisms will enter into the body, infect and cause death. So when many diseases are attacking a person, then the person cannot bear. For example, at a time when we are suffering with one disease like malarial fever, we are suffering a lot. At a time, imagine that in the person where HIV is there, where immunity level is lowered, 4 or 5 diseases are caused, then the person cannot bear it, then death is sure. That is, and it is causing the death. Diagnosis. Now that person who is having HIV positive, the incubation period of HIV is very long. For example, one day, today, one person is receiving this HIV today. Very next day itself, the person will not show the symptoms of HIV. Okay, the symptoms of HIV infection will be seen after 10 years, after 12 years. So during this period only, they are growing, multiplying, destroying the white blood cells. That is, it will take long time, 10 years. Now during this 10 years of incubation, the person looks normal, perfectly normal, normal and active. They will do the normal work, no fever, no severe symptom. So at that time, whether that person is HIV positive or negative, how to know that? It is very, very difficult. For that, some test we are doing in the laboratory. What is the test? First one is ELISA test. Now we can pronounce it as ELISA or ELISA, but the 5 letters must be written in capital letter because it is abbreviation. What is the expansion for ELISA? Enzyme linked immunosorbent assay. Assay means analysis. Enzyme linked immunosorbent assay. It is a procedure of blood test. It is a procedure for a blood test. So by that we can identify whether the person is HIV positive or negative. Now in this blood test, what they are doing? They are taking little bit of sample of a blood from the person who is suspected, and they are trying to detect the presence of antibody for HIV. Once a HIV enters into the body, immediately the blood of the person is producing antibody for whole HIV. Antibody will try to kill the HIV, but it cannot kill. The reason is outer envelope is there. I already told you, bilipid layer plus glycoprotein. They won't allow. Okay, so the virus will be alive. So if the antibody for HIV is there, the person is having HIV. If the antibody for HIV is not there, the person is HIV negative. HIV is not there in the body. Okay, the second test we are going to take, Western blot test. It is another one technique. It is another one procedure, Western blot. Now it is a confirmatory test. Now with the ELISA test, 99% we can confirm whether the person is positive or negative. Suppose doubt is there little bit. Now we can go for Western blot, which will 100% says whether the person is positive or negative. Now what it is doing is, in this test, we are detecting the antibody for viral core protein. Now what is that viral core protein? Core protein is the outer envelope where the glycoprotein is there. No? Then first our body will produce antibody for whole HIV. Okay, the HIV is not killed. Then our blood will take the second step. What is the second step? It will produce one more antibody, separate antibody for only the core protein. The outer envelope protein is there, no? It will try to produce for that. If the envelope is destroyed, virus can be destroyed for that. If the antibody for viral core protein is present, HIV positive. If the antibody for viral core protein is absent, that person is HIV negative.